IB revision video, Mr. Phillips, and today we'll be looking at homeostasis and blood glucose control. You can see here the risks of blood glucose, of medical issues, with different levels of blood glucose in your body. So you can see that there are dangers if you have low levels or high levels. Now, homeostasis is the maintenance of a constant internal environment, and it should be independent of the external environment. So here you can see with this bird that you have a high fluctuation of the external environment, but your internal environment doesn't change. If the temperature, for example, goes from 20 degrees one day to -5 the next day, your body temperature doesn't fluctuate massively as a result. Now, your internal environment, though, is constantly fluctuating around an acceptable range. So whilst I'll talk about precise glucose levels in real life, this isn't quite as accurate as it sounds. Now, homeostasis is controlled through a process called negative feedback. And this is the idea that if for example, your body temperature goes high, it's shown here, it's detected by sensors in your nerves and your skin, and it's sent to the impulse is sent to the temperature regulatory center in the hypothalamus, and that causes then sweat to be released, which causes your body temperature to lower down. So what's happened is your body temperature has gone high in temperature, there's been a change which restores the level or reverses that high level, lowering the temperature back down. So it's a process that reverses a change, taking the conditions back to their optimum level. The opposite of this is positive feedback, which is a mechanism that further increases the level. So an example that you need to know about this is in blood clotting. And here you see you get a break or tear in the vessel. Clotting occurs because platelets adhere to the chemicals, and it releases further chemicals which attract more platelets. The one that you really need to know in detail is the oxytocin example. At the end of pregnancy, oxytocin triggers contractions to start. That then is detected by stretch receptors, which trigger more oxytocin to be released, which triggers a higher concentration of oxytocin, which then triggers a higher level of contractions. And this keeps getting stronger due to more and more oxytocin until the birth of the child. Positive feedback is often a negative thing because it happens when homeostatic systems break down. So thermoregulation would be a bad thing because if you got too hot, you might get even hotter through positive feedback. So we're going to be focusing on glucose. So you get glucose from absorption from food in the digestive system. Glycogen, which is glucose stored away in the polysaccharide, can be hydrolyzed and broken down through enzyme reactions. And non-carbohydrates such as amino acids and lipids can also be converted to carbohydrates. The supply and demand can be very different, but glucose is normally about this level in the blood, and it's controlled by two types of cells in the pancreas. And the pancreas you can see here underneath the stomach. So the pancreas has two roles. It's got a exocrine role, which is digestive enzymes, but it has an endocrine role releasing two key hormones, insulin and glucagon, which we are going to look at today. The tissue responsible for this controlled blood glucose is called the islets of Langerhans, as said here. And it's made up of two cell types, alpha and beta. The islets of Langerhans, the alpha and beta cells, do different jobs. So the alpha cells, which are the bigger cells, release glucagon, and the beta cells, which are smaller, release insulin. If you want to identify the beta cells, they will stain dark with stains because that will be held onto by the insulin. Alpha cells release a hormone called glucagon that raises the glucose levels back up. So glucagon binds to a receptor on the cell surface of the liver cells only. This activates the phosphorylase of cytoplasm in the cytoplasm, which causes the breakdown of glycogen to glucose. This process is called glycogenolysis, and it increases the conversion also of amino acids to glucose via gluconeogenesis. So these two processes increase the production of glucose, which is then released into the bloodstream. When the blood sugar levels get too high, the beta cells release insulin, and insulin increases the uptake of glucose from the blood by its target cells. This is mainly in skeletal muscles and liver cells. They will increase their respiration rate to use that glucose up, and glycogenesis happens where glucose is converted to glycogen. So how does this work? How does this happen? Well, this is linking to receptors, which I've talked about in a previous video. What happens is kinase receptors are used. So you can see here that the insulin attaches, the tail of the kinase receptors comes together to make a dimer, and that triggers a series of reactions causing the vesicles in the membrane that contain these proteins called GLUT4 to fuse with the membrane. These GLUT4 proteins then allow glucose to enter via facilitated diffusion. So you need to be able to talk about this overall and talk about it through negative feedback terms. So when the blood sugar levels get too high, the beta cells detect it, release insulin, cause these changes to happen. When the alpha cells detect low blood sugar levels, they get activated, turning these reactions to happen. And therefore that increases the blood sugar levels or decreases them due to negative feedback. So diabetes is a chronic disorder caused by the inability to control your blood sugar levels, and it's generally caused due to two reasons: a lack of insulin or a loss of responsiveness to insulin. Its symptoms include high blood sugar glucose levels, hyperglycemia, presence of glucose in the urine, increased thirst and need to excessively urinate, and these can cause much more difficult issues longer term, including organ failure, uncontrolled osmosis. A glucose tolerance test can be used to diagnose diabetes, where they check your glucose levels after a certain amount of starvation. Type 1 diabetes is called either insulin dependent or early onset diabetes, and for this cause you're supposed to know it as early onset diabetes. This is when the pancreas seems incapable of secreting enough insulin, and generally it's due to the fact that the beta cells have been attacked by your own immune system, which is called an autoimmune disease. This is often onset at early age, so it's normally a few years old, and it is generally linked to a genetic inheritance disorder. Type 2 diabetes is late onset or non-insulin dependent, and this is where if we go to this one first, your receptor sites on the liver cells and muscle cells and fat cells fail to respond to insulin. That, though, may trigger your pancreas to release more and more insulin, and eventually your pancreas damages itself, so it may not release insulin either. This has a slow onset and may not be noticed for years. The risk factors linked to type 2 diabetes are obesity, a lack of exercise, a high sugar diet. It can be a family history, and the older people live, the more likely they are to have this. So its cases are increasing, particularly in the developed world. How can we treat both types of diabetes? Or type 1 diabetes, because it's due to a lack of insulin being produced, is generally treated by insulin injection shortly before meals. This is really important to time right, because the insulin is broken down in the blood, so it doesn't last long, about 20 minutes or so, so it has to be used at the right time. Stem cell therapy is also being looked into this to try and work out if we can use it to fix the pancreatic cells. Type 2 diabetes, you generally try and eat a healthier, balanced diet with lower sugar levels. You can eat small meals frequently to avoid peaks of sugar and troughs of sugar. High fiber in food in diet, so a lot of the fiber food doesn't get digested and it just travels through. Losing weight is obviously a big one. Regular exercise and glucose lowering medication.